Good morning, everyone. Thank you very much for joining us here for the second of our joint symposia between the National Biofilms Innovation Society and the India Biofilms Society. My name is Mark Richardson. I'm from the National Biofilm Innovation Centre, and today I'm being my colleague Snehal Kadam from the Pune University, and the India Biofilm Society is helping me chair this session. So this morning we've got 6 speakers. 3 who are part of the National Biofilms Innovation Society community, and 3 who are from the India Biofilm Society community. You will see as you've arrived that you are on mute and you don't have access to your video either. You do, however, have the ability to ask questions in the Q&A box, and we encourage you to do that. And if there is time during the session, we will take some of those live. If not, they will all be answered either during the session by the presenter or afterwards, and you will receive an answer. At the end of the session, the recording of this will be posted, and you will get a link to that as well. So you'll be able to watch any bits you were unable to see fully. And our first presenter this morning is Gavin Mello, who's from the University of Edinburgh and is an NBIC fellow. And he's going to be talking this morning about the soft matter physics of biofilms. So Gavin, I'd like to hand over to you, please, to give us our first presentation today.
Okay, so hi. First of all, I'd like to say thanks to NBIC and the India Biofilm Society for organising this and for giving me the opportunity to talk. As Mark said, I'm Gavin and I'm an NBIC fellow at the University of Edinburgh. And today I'm going to be giving you a little run through of what research we do at Edinburgh, particularly in the context of soft matter physics. Right. So what is soft matter? Well, basically soft matter is any material that is sort of squishy. So not quite a liquid, not quite a solid, but somewhere in between. All right. So some examples of this would be chocolate, paint, cornstarch. So let me show you a little video here. This is a classic experiment demonstrating non-Newtonian mechanics in fluids, where if you hit the suspension or this container of cornstarch with a bowling ball at a very high speed, it doesn't penetrate the fluid. So when it hits the fluid, it interacts with the fluid like the fluid's a solid, but if it was allowed to sit there, it would slowly sink to the bottom. So the mechanical response of the fluid really depends on the force that is applied. And when you look at these materials in more detail, what you'll see is essentially they are all systems of colloids. So colloids are usually micron-sized particles, and often they can cluster together and form aggregates and give rise to emergent physical behavior and non-trivial mechanical responses. And if you look in many colloidal suspensions, there are often polymers present that are much smaller than the colloids, and they mediate aggregation and a lot of the behavior in many different ways. So you may have depletion attraction, steric repulsion, and bridging interactions. What has this got to do with biofilms? Well, essentially a biofilm is a soft matter system. You have your cells, which are approximately 1 micron in length. So these are like colloids, and they secrete a diverse plethora of extracellular materials, such as proteins and polysaccharides, and they mediate aggregation and interaction with the surfaces in many different ways, right? And often biofilms can be characterized as viscoelastic materials as well. So they're truly in the realms of soft matter. So next I'm going to just give you a little flavor of some of the experiments and simulations that we do here in Edinburgh, probing not just biofilms, but mainly bacterial systems more generally. So researchers here have used light-powered bacteria to create patterns on surfaces with respect to bacteria that that swim in response to green light being shone on them. So here is a system of bacteria, and I'm going to play the movie, right? And in the background is just some non-motile cells. And then we will shine green light at it. and some of the cells will start swimming, and the green light creates a pattern in the background. And what we see is that the swimming cells create the University of Edinburgh logo. So this is pretty cool, and we can do more interesting stuff, where we can turn a smiley face sad. And in this case, the swimming bacteria here in the background are being treated with the light, and this pattern is from the non-motile cells, which there's no light there. We're also very interested in how bacterial colonies grow on surfaces. So this is an example of E. coli growing on agar, and how we can use the tools of soft matter physics, and in particular liquid crystals, to describe this phenomena. So in this paper here, the researchers looked at describing bacterial growth as an active pneumatic liquid crystal. And you can also imagine that in these colonies, if they were constrained laterally, what would happen is that the growing colony would make a transition into the third dimension. This is known as buckling. And researchers in our lab have also looked at this phenomenon. So here's some confocal microscopy images of E. coli, and it's growing radially, but it's constrained in the radial direction. And what we see is that there is a buckling into the third dimension in height. And the researchers have complemented these experiments with some very sophisticated agent-based computer simulation models and recapturing that behavior. We've also done extensive work led by Professor Kate McPhee on biofilm formation in Bacillus subtilis. So here is a Bacillus subtilis biofilm, and Bacillus subtilis forms these really undulating morphological biofilms that are very distinct. And what is unique here is that this biofilm is hydrophobic, all right? So if you were to take some water droplets, arbitrarily colored red, yellow, and green here, they don't wet the surface. What they do is they remain completely spherical. So the biofilm is hydrophobic, and this hydrophobic activity is because of a surface active interfacial protein called BSLA, which our group have done a lot of work on. We are also currently doing some work led by Dr. Aidan Brown on using bacteriophage to prevent biofilm formation on surfaces. So Aidan is doing this work with a company called Fixphage, and Fixphage are trying to treat surfaces with bacteriophage so that to prevent biofilm formation. So this is an electron micrograph of a glass surface, and these little bright spots here are the individual bacteriophage particles. Okay, and the SEM work was performed by Tom Glenn. And Rosalind Allen and Bartek Václav are using physics to try and understand the emergence of antibiotic resistance in using both theory and experiments. And this may be done in the context of fitness landscapes, antibiotic drug gradients, and how these affect colony morphology, etc., etc. So now I'm going to tell you a little bit about some work that I do specifically. So I've been very interested in biofilm formation and aggregation of Pseudomonas aeruginosa. Okay, so here is a confocal microscopy image of Pseudomonas cells on a glass surface. This is time zero in a flow cell biofilm experiment. So this is performed by my colleagues, Kasper Krag in Denmark. So we have individual Pseudomonas cells here, and in the center what we see is this clump of Pseudomonas cells. Now this clump was formed in the liquid media in suspension prior to inoculation on the surface. So what happens when this thing forms a biofilm? So 24 hours later, we see something like that. And we have done some work looking at the effect of these structures on the final morphology of the biofilm and the fitness of the aggregated cells in the biofilm. And to your right here, we've complemented these experiments with some agent-based computer simulations. Now, what I'm interested in now is how these aggregates of Pseudomonas aeruginosa form in the liquid media in the first place, in the absence of a surface. Okay, so I've done some experiments to look at this. So basically, we grew the bugs up, get them to some exponential phase, and we take a sample and put it under the microscope. And what we would see is something like this image here on your left, where we have these large aggregates of cells surrounded by a suspension of single cells. And what we want to do is try and characterize this aggregation behavior. So we then process this image to create a binary image like this on the right, where we have the aggregates and the single cells. And then we repeat this many times. And what we can do is we can build up a distribution of aggregate sizes. So in this graph here, what we see is how this distribution of aggregate sizes progresses with time. And what is important, the take-home message from this is that we can get a single cell peak here, corresponding to single cells in the images, and an aggregate peak here, okay? So armed with this sort of analysis, we wish to understand what polysaccharides are important for mediating aggregation in Pseudomonas aeruginosa. Now, from biofilm formation and biofilm studies on Pseudomonas, we know that there are polymers that are very important, such as alginate and PSL and PEL. But for this strain, which is PA01, it's PEL and PSL that are predominant for biofilm formation, but are they important for aggregation? And so if we repeat the analysis on some mutant strains, so this is the delta PEL, delta PSL, that produces neither polymer, we see there's no aggregate peak, and we have no aggregates in the system. And this is the same with the PSL mutant, which produces PEL, no aggregate peak, no aggregates in the system. But for the delta PEL strain, which produces PSL, we see that we can recapture the same behavior as we did for the wild type, right? So we have an aggregate peak and we have aggregates in the system. So this suggests that PSL is important for aggregation of Pseudomonas aeruginosa cells in liquid. But there are two types of PSL. There is a cell-free form in which the PSL is secreted away from the cell, so it no longer remains associated with the cell membrane. And there is a cell-associated form. So in this case, the PSL is synthesized but remains attached to the cell membrane. So we now want to understand which one of these, if not both of these forms of PSL, are at play during aggregation. So we came up with a little thought experiment that we then followed up with some actual experiments. So I'll take you through the thought experiment first. If we were to have a suspension of red non-producers, so these cells here, the delta PEL, delta PSL, and green producers, and we were to mix them up, what kind of aggregates would we see? Well, if it was the cell-free form, then we would expect the aggregating capabilities of the PSL to be shared amongst all the cells, and we might see aggregates composed of cells of both colors, red and green. However, if it was the cell-associated form, there may be two scenarios. You could have a non-specific interaction in which a green cell combined to a red non-producing cell, or a green cell could attach to another producing green cell. And in that case, we would also expect to see aggregates composed of both red and green cells. However, if it was some sort of cell-associated specific interaction, we would see that perhaps green cells only bind to other green cells. So producers only bind to producers. And in fact, when we did the experiment, so a big reveal here, but in B, what we see is that we have our aggregates of the Pseudomonas cells. And if we look in closer, we see we have these aggregates here. And if we switch on the fluorescence, indeed what we see is that the aggregates are only composed of green cells, which maybe supports this hypothesis here that it is cell-associated PSL that is mediating aggregation in the system. And we've also, in separate studies, tried to understand aggregation in these systems using particle-based computer simulations and field simulations. We'll see a little aggregate appear here. Right, now I'm going to talk about, for 2 more slides, a separate system.
Gavin, I'm going to ask you to wrap up.
Right, okay. So this is another system we're doing. This is Common dinitrificans, and basically it forms this pretty cool gel-like structure on the surface. We see all the cells are interconnected. And we do some experiments and simulations to look at that as well. So this is a bottom view of aggregating, sedimenting rod-shaped particles.
Wow.
And with that, I'd like to thank everyone whose work I have had the opportunity to present today. And in particular, I'd like to thank Kate, who's co-director of NBIC, one of the directors of NBIC at the University of Edinburgh, for funding this research. And thank you all for listening.
Thanks, Gavin, for a fascinating insight into the soft matter physics of biofilms. And Gavin, like all the other presenters today, is keen to reach out to other researchers who may want to collaborate or share experiences for future collaborations. So it's my honour now to hand over to our second presenter today, who is Dr. Campbell Gourlay from the University of Kent in the UK. And Campbell's going to talk to us about the detection and treatment of fungal biofilms on medical implants. Thank you. Thank you, Campbell.
Okay, thank you, Mark. And I'd also like to thank NBIC and all the participants viewing today. Good morning and good afternoon. So I'd like to really just talk to you about our work on fungal biofilms with respect to applications of those. So I work within the Kent Fungal Group, and this is a grouping of research groups, and we are all interested in different aspects of fungal biology. So some of us are interested in recombinant protein production and high-value therapeutics. Others are interested in using fungal cells to study cell biology. And we're also interested in pathogenesis, both of animals and plants with respect to fungi. If you're interested in the work we do, you can check out our website at the bottom there. Okay, so, oh, it's not progressing. There we go. So my particular lab's interests, we're interested in, we work with some physicists and some polymer scientists, and we're interested in developing inhibitors, modifying polymer surfaces to develop new materials that could be antifungals. We're also interested in developing new targets. And we're particularly interested in this with respect to biofilm formation on medical implants. And so what I'll talk to you today is about a little bit more about that. So we're really interested in how you can detect and manage microbial biofilms, polymicrobial biofilms, but driven by fungi in many cases, on human implants such as voice prosthesis or tracheostomy tubing, catheters, that sort of thing. And we're also interested, I'll just touch on this at the end, we're really interested in developing novel passive electronic detection systems using radio frequency identification, RFID, because this is quite a good technology to be able to use for detection when an implant is actually inside the host, inside the human body. So in vivo detection of biofilms on an implant when it's inside the host. So I'll just touch on a couple of these. I want to talk, we're really interested in trying to translate our research into the medical environment. And so I want to give you an example of how we've managed to do that. And that is with respect to, sorry, I'm having some problem with my slides progressing. This was with respect to this medical problem, and it happens in patients who have unfortunately had advanced laryngeal cancer. Typically, they would have their larynx removed in a procedure called a total laryngectomy. So their larynx would be removed, as we can see here, the larynx, and on the right we can see after the operation, the patient's larynx is gone. That means they've lost their vocal cords and they no longer breathe through their mouth. They breathe through a hole in their throat called a stoma. Okay, so these patients lose the ability to speak. They no longer breathe through their mouth. This is a life-altering operation, and it also means that they're very prone to infection, lung infection. So this can be, the speech can be restored using something called speaking valve or a voice prosthesis. And this is a small silicon rubber valve, a very simple device, a one-way valve. And here we can see the patient with the stoma after the laryngectomy. And what the valve does is it redirects air from the trachea. Instead of going out through the stoma, that will be covered, and the one-way valve will direct air through into the esophagus. And your esophagus does have enough vibrational capacity to be able to make a sound, which can then be, you can be trained by a speech therapist to manipulate that sound to speak again. So this very simple valve can restore speech in these patients. So it's really life-altering medical device for these patients. The problem that occurs is one to do with biofilms, because these medical devices, if you see where they're sitting, the valve is facing your esophagus, so it's constantly being bathed with food, substances, saliva, and the microorganisms that come from the upper respiratory tract and your upper esophagus, and any that are coming in from your mouth at all. So very rapidly, our prosthetic, this is a prosthetic removed from one of our patients after about 2 months, and you can see it's covered in plaques. It's covered in biofilm. These are polymicrobial biofilms, and what they do is they block the valve, so the air is no longer transmitted properly. The patients lose the ability to speak, and we have a nice reservoir of infection here that leaks through the valve into the lungs, and the patients get a lot of chest infection. So this contamination can happen very rapidly. 2 weeks is not uncommon. Certainly the average is 3 to 4 months for replacement of one of these valves. So the patient for the whole of their life has this problem. We wanted to understand that and come up with a plan that we could implement in the clinic. So to understand the nature of the biofilms, of course, we looked at these prosthetics, looked at the bacterial presence. So this is a graph from 159, early failing, so ones that are failing in less than 6 months. And we looked at the bacterial content, and you can see by far the most common bacteria that you get is Staphylococcus aureus, perhaps not surprising, and then Pseudomonas aeruginosa next. A lot of the things you'd expect to find that are commensal or commonly found in the upper respiratory tract in your mouth. We look at the fungal presence of these biofilms. It's all yeast. You just find yeast on these prosthetics, and again, unsurprisingly, the most common is Candida albicans. This is the most common human fungal pathogen, and it's highly prevalent in your upper respiratory tract and in your mouth. So the question really is, how do you approach this? Because we've got a lot of yeast and we've got a lot of bacteria and these plaques. What should we do? How should we treat it? So our approach was to try and see if we could tackle the fungal contamination, and we reasoned that might be successful because the fungal biofilm is larger, it's much more dense. It's the polysaccharide matrix they produce is very, very sticky, and so we thought this is a physical blockage problem, and we might be successful in reducing that by treating the fungal contamination. Anecdotally, our clinicians tell us that the plaques look like they're yeast plaques predominantly. And also there's some literature that suggests this might be a useful avenue to go down. There's something else that's really important with regards to biofilms that we've discovered, fungal biofilms, and this is the presence of high levels of carbon dioxide. So in your exhaled breath, you have about 5% CO2. That is a trigger in many of these yeasts, and in particular Candida albicans, for growth. So in Candida albicans, if high CO2 will force them to transit from a yeast form into this hyphal form, which is much more adhesive. So it sticks down and forms a much thicker biofilm. So there's a real trigger in your breath, and you can see this if you simply contaminate your prosthetic in air or in CO2, high CO2, 5% CO2, you can see macroscopically the increase in biofilm formation in these much larger plaques. So we've really drilled down to this in our research, and we understand the molecular basis. So we have a paper that's just in final revisions, but it's on bioRxiv if anybody wants to check that out if you're interested in this. So we're really interested in that. With our clinician partners, so we're part of multidisciplinary teams within the clinical environment. So we work with clinicians to implement our knowledge about biofilms to try and come up with treatment plans. And so we've just published a paper on the voice prosthetic. We formulated with our clinicians the best antifungal treatment strategy and tested that in patient cohorts. And here you can see the voice prosthesis lifespan before our treatment plan and then after our antifungal treatment plan. And you can see on average we get about a 270% increase in the lifetime of the prosthetic, which means patients who would have had theirs changed every couple of weeks now are maybe coming in every 6 months. This has been really good in the current pandemic because patients have a home management plan these vulnerable patients didn't need to come into hospital to get their prosthetics changed because they had our plan in place. So this has been quite successful. It's fully ratified within the UK National Health Service, and we have distributed the pathway to all of these trusts in the UK, and also it's now being distributed in Australia and in the US as well. So we've got some nice anecdotal evidence from our patients. So now it can be months before I have the prosthesis replaced, when it used to be weeks. The new pathway has been a game changer for me. So this is an example of what we try and do. We try and use our knowledge of biofilm growth and implement it into a clinical setting. Just a last couple of slides. We're really interested in trying to detect the biofilm growth within the patients using electronic detection systems. So we work with electronic engineers, and we build systems where we embed RFID detection systems into the polymer. So this is an in silico design of one of our prosthetics, and it has the electronic RFID sensor built into it. These are passive, battery-free devices, and what they're doing is monitoring the electrical properties of the surface of that device only when it's inside the patient. This is interesting. So the scenario is we have a clean prosthetic with our sensor inserted into the patient. It starts to get contaminated, and something happens on the sensor of the device that changes the electrical properties that we can detect outside the body. And with regards to fungal biofilms, it was interesting to hear the last Bacillus subtilis having has a very well known to have a very hydrophobic character. Candida albicans biofilms, for example, the electrical properties of their biofilms change through growth. So here we have a growth using a device that measures capacitance. And you can see that the capacitance increases in the early biofilm formation, it peaks, and then the biofilm becomes much more conductive, and the capacitance drops back down. So we can use these changes within the context of our electrical sensor devices and start to monitor whether our treatments are being effective, potentially, and even you build these into other devices that are more commonly just removed without any real monitoring of contamination, such as tracheostomy tubing. So this is an area we're really trying to develop further. I'd just like to thank, this is the members of the lab and some of the clinicians that are involved, and just also like to thank our funders, and some of our funding has come from NBIC, and I'd like to just leave it there. Thank you very much.
Thank you, Campbell. Thank you for sharing with us that excellent piece of translational activity around prevention and detection and management of biofilms in perhaps a neglected area. So thank you very much. I'm afraid we don't have a chance to take questions live, but I would encourage people who want to connect.
So Mark, I have a question for Campbell. So Campbell, you showed that Candida albicans turns to hyphal form on 5% CO2 exposure. So is it the case with when it is hypoxic? If you remove oxygen, will it do the same thing?
What hypoxia will do, hypoxia will change the cell wall structure. It won't induce hyphal formation, but the cell wall structure will change. Nobody has really investigated the significance of that with regards to biofilms yet, actually.
Yeah, because what I'm saying is we tried to infect catheters, central venous catheters with Candida albicans. We wanted to track using luminescence. And what we found is that with inside catheter, within 24 hours to like 48 hours, they were like all, Candida was like all hyphal. There was like a lot of hyphae, and it was like all clogged and blocked.
Okay, interesting.
So that's why I got like maybe it's, you know, the CO, instead of CO2, it's the less oxygen, because in CO2 incubator, when you put 5% CO2, you are actually lowering oxygen.
Okay, it's a complex question. There are a number of factors that stimulate morphogenesis.
Yeah.
So,
Ashwini and Campbell, could I ask that maybe you could correspond?
Yeah, definitely. Thank you.
Sounds like there's a real opportunity for discussion there. Campbell, could you stop sharing your screen?
I'm sorry, yeah, of course, yeah.
Thank you. And the final, so thank you very much, Campbell. And that was Dr. Ashwini, who's one of our speakers who you may want to connect with. And finally, I'd like to ask Dr. Miraz Rahman from King's College London, who's going to talk to us about the development of new anti-biofilm agents through the repurposing of existing licensed drugs. Thank you, Miraz.
Thanks, Mark. At first, I'd like to thank NBIC and the Indian Biofilm Society for giving the opportunity to present our work, which is partly funded by NBIC. So I'm a medicinal chemist and a drug discovery scientist, but I'm working in collaboration with Public Health England as well as University of Bath, where we are performing different part of the project. So we all know that it's quite urgent to find new therapeutics because AMR is on the rise in the clinic, and biofilm-associated infection is one of the main cases where the treatments are failing. Now, as a drug discovery scientist who have been involved in setting up spin-out companies, it's very difficult to attract funding in this space because of lack of commercial success or commercial interest. So repurposing drug can be one attractive option because in normal cases, it takes a very long time and huge expense to identify a new drug which can be taken to the clinic. But in case of repurposing, it skips many stages in the drug discovery process if it can be done properly, particularly when we are looking at agents which has been already approved or where we have got the known safety profile, known pharmacokinetic profile. At the same time, sometimes repurposing candidates provides new chemical scaffolds that may not be optimum to be taken directly to the clinic. However, that can be further optimized using medchem approaches, and then you get a head start in identifying a chemical scaffold that is much more druggable. So we have, as a medicinal chemistry group, as part of our work, we also do curate a repurposing drug library that's been assembled from getting information that's available in the public domain as well as our own work. We have got about 15,000 members, and we would be happy to collaborate with anyone who has got a particular target where we can screen our compounds against the target and relevant model to take them forward. And all the compounds have got known safety and PK profile. And we only include compounds that have been either marketed or progressed to phase 2 clinical trial. That means we know they are safe to profile. We can dig out all the pharmacokinetic profile. We know their dose regimen as well. In some of the libraries that is available in the public domain or from other commercial partners, sometimes it's very difficult to identify compounds that you can obtain from the commercial vendors. So what we do that we continuously check about that and only keep compounds that can be directly purchased from commercial vendors, because sometimes the compounds that have been marketed is very easy. Either you can work with the companies, or most cases, suppliers like Sigma or Fluorochem, you will be able to get them. But the major difficulty is where you have got the experimental drugs that may have progressed to phase 2 clinical trial, but then didn't progress. And then sometimes getting hold of those compounds can be quite tricky. And we group them into different therapeutic categories. Now, within the research group, efflux is a key research interest. We have been looking at how antibiotics or antifungal agents, they get effluxed, and how different substrates and inhibitors behave differently when they interact with different efflux pumps. We have got quite a powerful supercomputer system within our group. As we probably all working in the antimicrobial space, we know that there are very few crystal structures of efflux pumps. But we have got the system to develop efflux pumps from using homology modeling and then run microsecond to millisecond simulations to validate some of the research findings. And what we have done using that knowledge, that how different compounds interact with the pump and how pumps are able to efflux out most of the substrates, but some inhibitors can selectively block them. We developed a technology which is called efflux resistance breaker technology, and King's has filed 2 patents. One around, in general, the concept about antibiotic resistance breaker using efflux resistant breaker technology. And we have also generated new efflux-resistant antifungal agents, which works against all MDR Candida albicans or Candida auris. So again, looking at Campbell's work, we would be very interested to see whether we can find some common research interest. Now, as efflux is a key research interest within the group, we have been looking at how efflux plays a role in biofilm formation. We have done quite a bit of review and published. One of them, which has been published in Journal of Antimicrobial Chemotherapy, has been quite widely cited as well. We were trying to identify that how inhibiting efflux can prevent biofilm formation. And there, the research interest of one of my key collaborators, Dr. Brian Jones from University of Bath, where he's interested in looking at Proteus mirabilis biofilm. This formed crystalline biofilm in catheter, and the gene and the efflux pump that we have identified. This is a MFS type pump, that BCR-CFLA, and it plays a key role in forming biofilm. As you can see from the slides, in the wild type, the amount of calcification and the crystalline biofilm formation, and compared to the mutant at different sections of the catheter, is statistically significant all the way through. So we did a pilot project, and using a smaller library, we managed to identify two compounds. One is fluoxetine and one is thiodarzine, that inhibits efflux, and it attenuates the crystalline biofilm formation in Proteus mirabilis. And these compounds fit quite well within the efflux pump, and they were able to inhibit efflux and subsequent biofilm formation. But as you can see from fluoxetine, it's not probably the right therapeutic class which can be repurposed. So what we have done now, a project with support of NBIC, we are widening these and screening that 15,000 member library to identify more suitable therapeutic agents that can be taken forward. Now, I wanted to show this slide just to highlight that the problem, why this is an unmet clinical need. We know that in catheter-associated UTI, you will be using these catheters, and they are normally in place for a long time. And then you can see that the type of biofilm that forms. And when you are trying to remove this, the pain that the patients go through, and the infection from there, it's one of the major area where we need effective agents that can be locally delivered or used in combination with antibiotics, which can prevent this biofilm formation. We know that E. coli is one of the major pathogens in UTI, but we always find one of the major one is also Proteus, and it has got a strong ability to form biofilms. These catheter surfaces are almost ideal surface for these biofilm formation. So the project that we are currently working on, and again, this is really important, that where we have got a defined target and associated models to validate when we identify compounds through our in silico screening. So we are screening this 15,000 strong library. Our main goal is trying them directly into these in vitro efflux assays. For those who are not familiar with these assays, this is how we use a fluorescent dye to measure whether a compound can prevent efflux. So normally when the efflux pumps are operating, the fluorescent dye gets in and then gets out, and you don't really see fluorescence within the bacteria. However, when you have got an efflux pump inhibitor which can block the pump, you start to see accumulation of efflux, accumulation of fluorescence within the bacteria, and that can be quantitatively measured. So we would like to take compounds, drugs directly unmodified, but in some cases, we are also doing some medchem optimization and then putting it through the efflux assays. And then we have, this is the model at University of Bath, where they have got this in vitro bladder model, which simulates the environment where the catheters will be in case of UTI. And we would like to see the compounds that progresses through this in vitro efflux assay into in vitro bladder model, and then only the compounds that significantly works within that in vitro bladder model will be taken forward for pharmacokinetic assessment and further efficacy. So I will just very briefly go through the protocol that we use, that we normally select about top 600 candidates, and then you have to start to apply filters, because initially you don't apply any filter. But depending on your therapeutic indication, you have to apply toxicity filter. For example, anti-cancer compounds needs to be eliminated, because in this case, like biofilm, where you will be using potentially a large dose over a period of time, you cannot really use these toxic compounds. And then dose filter, like most anti-infectives are high dose therapeutics, so you have to select compounds which are safe at a relatively high dose. Similarly, we apply the PK filter. For example, in this case, we are only selecting compounds which are at least 25% eliminated unchanged via urine. That gives you the opportunity to interact with the catheter and the positive agent so that it can prevent the biofilm formation. So by applying different filters, we finally select about between 50 to 100 compounds candidates for in vitro screening. So I cannot share because of the IP reasons the results. We have got really exciting results for some of the compounds with therapeutic classes, which we think that we are safe enough and the doses that are used are high enough to be taken forward. So now these compounds are being validated using the in vitro bladder model. The COVID struck, so the work which we were doing in silico was much easier. Some of the lab work we have now resumed. So we will be hopefully by the end of this year, we will have some interesting result that we can publish. Now, as we have said, we are part of a collaboration with Public Health England and University of Bath. And what I would like to share with you here is an open innovation approach that we are partnered with Public Health England, which invites partners from across the world to submit compounds that will be screened for free and after suitable agreements. And the goal of this is to identify compounds that particularly works against either gram-negative bacteria, but we also have got antifungal panels. And one of the points that I would like to highlight that although it's a bit farther down the pipeline is where we are testing compounds against single and mixed species biofilm assays. So if you are interested in submitting your compounds and getting the results back within a reasonably quick time, and we also have a mixed model with Public England where the scholars can come and work with them while they are performing the screening, you're very welcome to contact. So finally, I would like to thank Shirin. Shirin is one of my postdocs who has been with me for a very long time, and she does all the in silico part of the work. And Mark Sutton is the microbiology team leader at Public Health England, who is a key collaborator, and Brian at the University of Bath, who has these in vitro blood model. And thanks for listening. And I'll be happy to answer questions that you post through the chat and reach out to you. Thank you.
Thank you, Miraz. And yes, please do post questions in the Q&A box for Miraz to answer. You can answer those live on the box now or contact him later. So I'd like to now hand over to Snehal Kadam from Pune University to introduce our three speakers related to the India Biofilm Society. So over to you, Snehal.
Thank you so much, Mark. Our first speaker is Dr. Ashwini. So Dr. Ashwini, you can maybe just start sharing your screen.
Yeah, sure.
Yes, so Dr. Ashwini is from the Tripura University in India, and he is going to be talking to us today about eradication strategies for catheter-associated biofilms.
Okay, thank you. First of all, thank you, Mark, for agreeing on conducting this beautiful symposia where we can interact. And good morning, everyone in UK, and good afternoon, everybody in India. So as we all know that we are talking about biofilms, so I won't go into the details of this, but if you see the left side, you can see the crux of our lab, what we are doing. So this is the central venous catheter, which is isolated from a rat after 10 days of infection in Staph aureus. You can see this is how biofilm is formed, and you can imagine how it's going to impact the host. And below that is a CVC, which is coated with a FDA-approved polymer of methyl cellulose, and you can see that how well it is protected. And only you can find inside there's a little bit of tissues and cells. So today I'll be talking on central venous catheter associated biofilm infections and the strategies that we use in the lab to eradicate, whether preventive or curative. So we all know the biofilm, what are the biomedical complications associated with the biofilms. We know that there are persister cells which are tolerant to antibiotics, recalcitrant to host difference. They cause a bunch of diseases from your head to toe, and all the prosthetics or devices that are implanted get infected with biofilms. So we mainly concentrate on central venous catheters. And of these central venous catheters, we mainly concentrate on totally implantable venous access ports because these are like new, these are safer in cancer patients, used in hemodialysis patients, parenteral nutrition, and critically ill patients. But it's well documented that 15 to 20% of these get complications associated with biofilms. They get infected. And the plus point of using these for studies is that these are totally implantable. These are closed chamber, but still have access to the host system. So that's why we selected these. So our lab focus is, as we say, that we are like interested in chronic biofilm associated infections, and the model system that we use is central venous catheter, which is TVAP or POTA-CATH. And the organisms are Pseudomonas gram-negative as well as gram-positive. And this is based on the clinical data that we got in France during my postdoctoral work, that it's predominantly infected by Staph aureus and Staph epidermidis, which is unlike in India, we normally find gram-negative bacteria. And I have started collaborating with Anusha, who is here, who's a clinician. So the approach that we use is we try to understand how host pathogen and device interact. So this trilateral interaction between these three partners which are associated when you study biofilms on devices. So we think that this is a more holistic approach, because if you neglect one, you are ignoring that partner in this interaction. So we are a very traditional microbiology lab where we use routine microbiology, culture techniques, microscopy, molecular, biochemical, and physiological techniques. I wouldn't go more in details on that. So what we have developed is that once we do these 96-well static culture assays, we go into in vitro continuous flow system that we have developed and it's published, where we use the live device. And we can control the flow using the peristaltic pump. So what we can do the controlled biofilms can be grown in the devices directly. And you can assess the eradication preventive strategies as well as whatever molecular mechanisms that you are interested in. So we study using this. However, the host factors are mostly missing. In most of the work that is done pan world is like in vitro, where the host factors are missing, and the in vivo models are not there. Not very many in vivo models for biofilms are there. So we developed this in vivo model, animal model. This is what I'm going to present to you. So here what you see is a totally implantable venous access port, which can be implanted subcutaneously under the skin, and we use rat. As you can see here, this is the port which is implanted under the skin on the dorsal side of the rat, and then we tunnel the catheter under the skin and implant it into the jugular vein of the rat. So once this is done, what you can do is you can infect the rat, the lumen, using the Huber needle. Huber needle is a specialized needle to enter through the septum of the port. And this way, this allows you to make at least 1,000 to like 1,500 punctures without damaging the septum. So you can use light if you want to follow the imaging, or you do not have to particularly use the light, as we do not have imaging system here in my university at the moment. So this catheter model, you can monitor non-invasively if you have a luminescence bacteria. This is from my postdoctoral work at Institut Pasteur. This is where I developed this model, and we are still using it. So it forms a chronic biofilm, and you can monitor biofilms inside the catheter up to 120 days, even in fact more than that. But normally for a routine experiment, what we do, we just euthanize the rat on day 10, and we do the microscopy, and you can do the microscopy to confirm the biofilms, and quantification is really important to make sure that this biomass is proportional to the number of bacteria that is in there. And this is the catheter harvested on day 10. So how do we use this model? So we have like different things going on in the lab at the moment. So we use the rat, take out the catheter, and then we take out the biofilms. We want to study the new properties, because biofilms very less is known about what are the properties in biofilms and how we can utilize these properties. So we take out these biofilms, study them, and utilize them to understand what are the eradication strategies that we can build up, the anti-adhesive properties, if we can prevent the formation of biofilms on catheters, and we can go back into the rat model to evaluate it. And then we are interested in finding diagnostic markers, because diagnostics of biofilm is very challenging. We all know that it's earlier the better. And the early markers are missing, so we want to find the early markers of biofilms. And since we have a rat model, so it's a very good model. We have done a lot of things with it. You can go and read our nature protocols, it's published data. And we have done all the things that doctors would do in clinics with the patients. So everything is done in here. So another thing we are interested in to understand how host and biofilms interact, because most of the things that are done are in relation to the planktonic bacteria that the host system is able to kill. But as soon as the biofilm comes, host system is not able to understand what actually it is. It's new for it. So because we do not know, it's not new for the whole system, but for us as a scientist, as a researchers, it's new. So this is what we are doing in the lab at the moment. These are the questions that we are trying to address. And one of the projects that is going on is to develop, we are trying to explore the nanomaterials, if we could coat them on medical devices. It's funded by Department of Science and Technology, Nanomission project, and by Indian Council of Medical Research. Another project is funded by UGC, where we are trying to explore, since I'm in northeast of India, and India has 5 major hotspots where a lot of diversity, floral diversity is there, and a lot of the local tribal people in Tripura, they use a lot of medicines which are local, and they're dying. The information is dying. So we want to explore the traditional medicinal plants and see if we could target persisters and quorum sensing, and then evaluate in our model system. And we want to understand the mechanism of how these traditional medicines can help us. And then we want to try, we want to make antibiotic lock therapy. The clinicians will understand better what antibiotic lock therapy is. So it's a lock, the porta cat that we use. Normally in clinics, clinicians, they inject heparin to block, to prevent the clotting inside the catheter. And once the biofilm is formed, in case there is infection, they treat directly on the biofilm. They inject high dose of up to 1,000 times the MIC of antibiotic inside for 12 hours, and they let it dwell for 12 to 24 hours. So this is the antibiotic lock therapy. So directly treating. So we want to develop these things. So that's all for, this is what we are trying to do. And like I said, this is the focus of our lab. We are trying to understand the chronic biofilm associated infections and try to develop the strategies against biofilm to combat the biofilms. And we are open for all sort of collaborations. From we are collaborating with pharma people, physics people, all sort of people. And at the moment, what we are looking for is somebody who can do mass spec for us specifically, and RNA-seq to elucidate the mechanism of all the herbal compounds that we have. That's what we are looking forward for. And thank you. And this is where we are located at the moment. This is Tripura, a very small state on northeast of India. This is where Karishma is, and we are on the other side, Anusha on the south. So thank you. Thank you so much. Any questions if anybody has?
Thank you so much, Dr. Ashwini. I think if there are questions, you can just type them into the chat window. And I will now ask Dr. Neha to start sharing her screen. So our next speaker is Dr. Neha Jain, who's from the Indian Institute of Technology in Jodhpur. And today she's going to be talking to us about novel strategies for combating biofilm infections. Neha, you're on mute. We can't hear you. No, you're still not audible. No, Neha, we still can't hear you. Neha, do you want to take a few minutes to figure this out? And do you want, is it okay if I move to Sriram? Okay, all right. So I think we'll come back to Dr. Neha. So let's move to Dr. Sriram, who is from the Institute for Stem Cell Research, for Stem Cell Science and Regenerative Medicine in STEM in India. And he's going to be talking to us about yeast biofilms and metabolic plasticity.
Can you guys hear me? Sorry.
Yes, yes,
Okay.
we can, Sriram.
Yeah, so I'll take it away. Thank you guys for having me, and yeah, thank you guys for having me. I hope you can see my entire presentation. So my name is Dr. Sriram Marahan, and I am from the Institute of Stem Cell Science and Regenerative Medicine in Bangalore. And we are a multidisciplinary institute who does a lot of research pertaining to stem cells, but also other cool stuff. So to keep it short, I'm particularly interested in understanding microbial biofilms all along, and starting from my PhD where I worked on bacterial biofilms. I've now transitioned to understanding fungal biofilms, and I'll just give you a very, very short spiel of what we have done in the past 3-4 years at InStem. So basically, there are several cool aspects of biofilms, and one of the aspects that I'm really interested in understanding is how do cells that are isogenic in nature and that are growing in the same environment, they actually transition into phenotypically heterogeneous cells. And this is one of the hallmarks of many, many biofilms that we have seen so far in these 2 sessions we've had. And there are really good classic examples. For example, if you take the social amoeba dictyostelium, it actually exhibits beautiful phenotypic heterogeneity where under specific nutrient starvation, it can actually transition from normal slime-like cells to these beautiful stalks that actually have different types of cells that do different functions. Other cool examples exist, for example, in Myxococcus, where normal vegetatively growing cells can exhibit swarming, aggregation, followed by fruiting body formation. And all of these are classic examples of groups of cells that actually exhibit phenotypic heterogeneity, which is differences in phenotypes and differences in function, although they are all genetically identical. So the question that I was interested in when I started my postdoc at InStem was, can we understand this from a metabolism perspective? And the reason why I wanted to understand this from a metabolism perspective was a lot of times biofilm formation is driven or triggered by nutrient limitation, and metabolism is actually at the heart of nutrient limitation. So the model system that I use to ask this question is Saccharomyces cerevisiae or Baker's yeast. And certain wild strains of Saccharomyces cerevisiae actually form these beautiful rugose colonies under low glucose conditions specifically. I hope you guys can see my pointer moving. So when we actually took a close look at these rugose colony biofilms, we actually noticed that there was a lot of these architecture, and you actually could see these interconnected networks that actually ran across the circumference of the colonies. And because we actually saw these colony biofilms in low glucose conditions, we first wanted to understand what the metabolic state of these cells are. And these cells being in low glucose conditions should obviously perform this metabolic pathway called gluconeogenesis, which is the exact reverse of glycolysis. And we actually designed a fluorescent reporter to actually look at gluconeogenesis across this colony. And although our expectation was that all the cells will be gluconeogenic, we actually were very surprised that only some of the cells in these colonies exhibited gluconeogenesis, while the cells that are at the extreme periphery of the colony, they actually were not gluconeogenic. And we did a series of experiments to find out that these cells were actually glycolytic in nature rather than being gluconeogenic, and they exhibited high pentose phosphate pathway activity. This work is actually published in eLife, and you guys can take a look at it. And send me questions. I'll be more than happy to answer it. But what was striking to us was how are cells growing in low glucose conditions exhibiting a glycolytic state, which actually requires the presence of a lot of glucose. And this was really amazing for us. And using a series of analytical experiments using mass spectrometry and computational modeling, we came to find out this sort of new phenomenon called threshold dependent phenotypic switching, wherein cells can actually be present at a particular metabolic state at the beginning, and as they actually grow and develop into these biofilms, they actually produce certain types of metabolites, and these metabolites can actually reach threshold concentrations. And when this threshold concentration is actually achieved, you start to see synchronous switching of cell states to an alternate metabolic state, which can now consume this metabolite that is actually reached threshold levels. And this actually was the driver for this sort of phenotypic switching. And so now you have two cell states. One is actually gluconeogenic and one is glycolytic, although this is really unexpected in the conditions where these biofilms grow. So this was one of the early stages of our project. And once this got published, we actually wanted to go in a little bit deeper and try and understand as to how you are actually making these critical metabolites that actually result in phenotypic switching to an alternate metabolic state. And that is where this news story comes in, where we found that this metabolic pathway that I had mentioned before, gluconeogenesis, seemed to be very critical, because when you actually look at these wild-type biofilm colonies grown in low glucose conditions, and when you actually genetically knock out these gluconeogenesis pathway, you see complete loss of colony rugosity and the ability to form these biofilms. And we actually quantified it using the fluorescent reporter I showed you. So these colonies actually do not have these glycolytic populations, and they also actually lack the gluconeogenic population because they can't do the gluconeogenic pathway. And what we discovered was an underappreciated role of amino acids, and in particular this amino acid called aspartate. So what we know about basic eukaryotic metabolism is that amino acids can actually not only act as nitrogen sources for synthesis of nucleotides, but they can also provide carbon precursors for metabolic pathways like the TCA cycle, and that in turn can actually fuel gluconeogenesis. So we actually tested this hypothesis, and as expected, when we put these biofilm forming yeast colonies on a plate without amino acids, they actually did not form biofilms. And you can simply add back these amino acids and actually get these rugose biofilms back. And of all the amino acids that we independently tested, we actually found out that aspartate alone can actually rescue biofilm formation in these yeast strains. And of course, this was not going to happen in the absence of gluconeogenesis. And we validated this using our fluorescent colony reporter. And what we also found out actually was that the levels of trehalose, which is the critical metabolite I showed you a couple of slides back, was also actually affected in the absence of amino acids. And you can actually add back these amino acids or just aspartate and rescue trehalose levels. So this was pretty satisfying for us. And we actually did this carbon flux mass spec experiment to show that the carbon of aspartate can be traced all the way to trehalose. And I believe this is the first paper to actually show fully that aspartate can actually functionally provide carbon for development of gluconeogenesis. So this was all very interesting, and we actually, like I said, used agent-based model to actually understand budgeting of this aspartate resource. And what we found out, and I will go very quickly, is that this aspartate is actually budgeted both as a carbon source and a nitrogen source in the gluconeogenic cells, while the glycolytic cells use the aspartate as a nitrogen source, which is the conventional role of aspartate. And we can actually vary these carbon nitrogen budgeting in these models, and we can actually show that this is indeed true. And when we actually wanted to experimentally test this, we used this the power of mass spec again, where we use carbon labeled aspartate and nitrogen labeled aspartate, and we did some flux labeling experiments. And what we showed was that this carbon flux was much more higher in the glycolytic, the gluconeogenic cells compared to the glycolytic cells, while the exact reverse was actually seen in the glycolytic cells. So this is what we have so far. So we have demonstrated that metabolites can actually be plastic, and they can be used in different ways by different cells. And this is sort of a driving force for phenotypic heterogeneity in these yeast biofilms. And what is the advantage of actually having two metabolic states? And I'll I quickly have a couple of slides. So what we showed was that these gluconeogenic cells were more resistant to stresses like desiccation and freeze thaw, which often biofilms face when they are actually growing in the wild. This is particularly true for yeast biofilms or Saccharomyces biofilms that generally grows on tree barks. And while this is the advantage of actually having a gluconeogenic population of cells, when you actually have a glycolytic population of cells, you are actually able to efficiently expand and forage for nutrients. And when you disrupt this ability to actually have glycolytic cells, this advantage is sort of lost. And we also know that these glycolytic cells allow for directional foraging towards the source of nutrient, wherein you can actually see that in these wild-type biofilms, the region of the colony that faces the glucose source is able to directionally proliferate much more efficiently compared to the mutants that actually lack these glycolytic cells. So this is sort of the current model we have. So we actually have these yeast biofilms that exhibit metabolic heterogeneity, and this is driven by two primary metabolites, which is aspartic acid and trehalose. And in the beginning, aspartic acid acts as a carbon source and fuels the production of trehalose, while in the latter stages, once you actually have the emergence of a glycolytic population, it actually switches and acts as a nitrogen source and allows these cells to rapidly proliferate. And each of these metabolic states confers distinct advantages to the colonies, and this actually allows the colonies as a whole to flourish in a glucose-limiting environment. With that, I would thank my mentor, Dr. Sunil Lakshman, who's been of great guidance and support, collaborators from the Bangalore Biocluster, which comprises of INSTEM, the NCBS National Center for Biological Sciences and CCAMP, and my funding sources. And thank you to the NBIC and India Biofilm Society for having me. With that, I'll take any questions. Thanks.
All right. Thank you so much, Sriram. So you can check the Q&A box if everybody has sent you questions. So Neha, can you just unmute and check if your audio is working? Yes. All right.
Great.
Yes. So you can go ahead and
Let me
share
just start
your
my
screen.
sharing. Yeah. Is my screen visible?
Yes. Yes, it is.
Okay. Okay, I sincerely apologize for this mess up. My laptop doesn't like Zoom, so my students came and rescued me. Hi everyone, I'm Neha Jain. I'm at Indian Institute of Technology, Jodhpur. I'm a very new faculty here. And as Dr. Ashwin mentioned that he is in the extreme east, I'm in extreme west, and we are in the desert part of the India. However, this is the IIT Jodhpur campus, which is lush green, so it's kind of deceptive. So we have a very beautiful campus, and those who are in India are most welcome to visit the campus. So I'm going to present that what my lab would like to pursue in future. As we are a very new lab, we are just going to start the work. And due to COVID, we haven't started yet. So we are specifically interested in the particular kind of biofilm and the particular part of the biofilm, which is amyloid. So this is a rugose colony of a bacterial biofilm, which is developed in a lab. If you look into the details of this rugose biofilm, you will find that there is a lot of cells. And you can see there are fibers around these cells. So these fibers could be cellulose and proteins. So if we look close into these fibers, you can see this is an E. coli cell. So this E. coli cell is surrounded by a lot of fibers here. Now, if you take a look close into these fibers, we can see that these proteins actually polymerize on the cell surface and they make these fibers. So we are interested into these fibers and how these fibers contribute to biofilms. And if we remove these fibers, can we inhibit the biofilm formation? The beauty of this protein is that it is synthesized in the cytoplasm. It moves to the periplasm, where it keeps itself in a natively unfolded form, and then it moves out through a very small pore and assimilates as fibers on the cell surface. There are a lot of accessory proteins which are required. Now, in E. coli, this aggregated protein is called as curli, and they are called as amyloids because amyloids traditionally were found as a result of a misfolding of protein. So any protein that goes into a misfolded structure can make into amyloids. These amyloids were initially involved in different diseases. However, Professor Matt Chapman at the University of Michigan identified that bacteria are very smart, and then these bacteria can utilize this very rigid fold of protein to protect themselves from the external environment. So he identified that bacteria can make these amyloids. These amyloids become a part of biofilm and provides rigidity and resistance to external environmental conditions. What is the beauty of these proteins? The beauty of these proteins is that you can recombinantly express them in your lab environment. You can monitor that how these proteins move into an amyloid structure, and you can actually follow the entire path. Why this is important? Because if we can follow this path of making amyloids, we can also look into the inhibitors of these amyloids. That can we use anything that flattens out this curve? So basically, we can use any kind of a dye like thioflavin T to monitor this amyloid formation. And if we use any kind of inhibitor, this curve flattens out. So that's why these proteins become a very important part in understanding the biofilm formation and their contribution to resistance. Now, my lab at IIT Jodhpur is interested in understanding the formation of microbial amyloids and their role in biofilms. We would like to develop strategies to combat amyloid-derived biofilm formation. And if we could successfully derive these strategies, we would like to formulate those strategies and inhibitors that can be coated on catheters. An offshoot of these studies would be to understand the crosstalk between human and amyloid-derived biofilms. So humans also make amyloids, and these bacteria that we have inside our body also make amyloids, like gut microbes, they make amyloids. So we would like to study whether there is a crosstalk between human amyloids and bacterial amyloids, and whether this crosstalk has anything to do with the non-infectious diseases like neurodegenerative diseases. Now, to answer these questions, we follow very basic strategies to study the biofilms and bacterial amyloids. So we plan to use different techniques like fluorescence spectroscopy, biochemical and biophysical techniques, microscopy, cell culture based assays, and animal models. And I'm really thankful for Dr. Ashwini's talk, because then now I think we have somebody with whom we can collaborate for animal studies. And using all these techniques, we would like to get some of the answers that can we figure out some anti-amyloid therapeutics, which are very novel and unconventional. Can we figure out a putative role of microbial amyloid in non-infectious disease? And there are many microbes that make these amyloids which are still unidentified. So can we identify those microbial amyloids in biofilms? So what are our specific approaches? We would like to develop chemical chaperones and protein chaperones. So just to give you an example, we would like to have proteins that resemble E. coli periplasmic proteins. So as I mentioned earlier, that the protein that makes the fibers has to pass through periplasm. The periplasmic proteins keep that protein in an unfolded structure, so it doesn't aggregate. So those periplasmic proteins can be a hint to identify certain proteins in humans that can actually inhibit this amyloid formation. And once we identify these human proteins, we would like to synthesize peptides out of it, and we would also like to test some of the small molecules that can either individually or combinedly can control the amyloids and ultimately can control the biofilm formation. So just to give you an example, there is one periplasmic protein in E. coli, which is called as CSGC. So this is a highly beta sheet structure protein that stops the unfolded forming of the amyloids in the periplasm. Now what we did is we tried to figure out that we humans, do we have certain proteins like CSGC? So we found that there are a couple of proteins which have structural similarity to CSGC. However, there is no sequence similarity. So one of the proteins is transthyretin. So we've moved forward with this protein, and what we figured out is that if you add this transthyretin in an in vitro amyloid formation condition, here you get curl-like fibers if you don't have transthyretin, whereas you don't get curl-like fibers if you add transthyretin. In the cellular model, what we figured out that in the pellicle biofilm formation, pellicle biofilm is just like a creamy layer on top of a liquid surface. So here you see a pellicle formation, which is curl-like dependent biofilm. When you add this protein, there is no pellicle formation. And we took these cells under transmission electron microscope, and we saw that the fibers were formed when you don't have the protein, human protein, and when you have the human protein, the curl-like fibers vanished. We also took these studies into organism, and we found that if you delete this transthyretin, if you have a knockout of transthyretin, the load of organism increases much higher. So this is just an example to show you that the human proteins can actually act as a biofilm inhibitors. And these proteins must be much better than small molecules or other peptides, because these are the host proteins. So we hope that these proteins won't evoke any immune response. So what we would like to do further is to identify that can we have a signal peptide sequence with these kind of proteins, which are beta sheet structure, and can we engineer them so that these proteins can be, or peptides can be secreted out. And we would like to make more efficient inhibitors like CSGC and transthyretin in future. And to do these, our lab is actively seeking collaborations with people who have mutual interest. We would definitely like to have a lot of bacterial strains to try out whether these bacteria can make amyloids and can they contribute to biofilm in different conditions. And we are looking for a facility for imaging and quantify biofilms, because we don't have that facility as of now. And last but not the least, our entire group loves to talk to people in general, in public, and of course in the scientific discussion. So we are very open to public outreach. And this is my amazing group whom I haven't seen for last 6 months. And these guys, they just came back today and they actually helped me figuring out how to set up this. These are the certain fundings that I have received, which helped me to establish my lab. So we have very basic instruments, but anybody who would like to use these instruments are more than welcome to use. And thank you so much for your time. And again, I apologize for the disturbance. Thank you. I would like to take questions if there are any.
Thank you so much, Neha. Yes, you can check the chat window for questions. And I think that wraps up our speakers for this session. So I think I'm going to hand it over to Mark to finish up with some closing remarks.
Thank you, Snail. So thank you all very much, our 6 presenters this morning, for your interesting, exciting and well-targeted presentations. To all our attendees for joining us today, we thank you as well. Squeezing this into what is probably a very busy day for you today. Just to remind you that there is Q&A box if you wanted to put in any final questions, or to email either the India Biofilm Society or NBIC if you want to have any follow-up whatsoever. We will send out a link to the video, so you'll be able to watch this again or share it with colleagues who were unable to attend today. So thank you all again, once again, very much for attending, and have a great day and remainder of the weekend. Thank you very much.